sir, is that my screen is visible? Yes, sir. Yes, sir. Yes, good evening to all. A special thanks to the organizer for organizing this World Record Series. And thank you for the opportunity given for me. Thank you, sir. So I am today going to share our views and thoughts about the role of enzymes in recombinant DNA technology. So is my slide visible? Okay, okay. Yes, before going to the role of enzymes or the enzymes involved in recombinant DNA technology, we will discuss about the basics that is required for recombinant DNA or genetic engineering process. The basic thing that is required is a DNA. So we all know DNA, it is nothing but it is a deoxyribonucleic acid. It's a very large molecule. It is made up of smaller units called as nucleotides. The smaller units that is present in DNA is called nucleotides. And each nucleotide has three parts: a sugar molecule, which is a ribose or a deoxyribose, a phosphate molecule, and a nitrogenous base. The nitrogenous base is a part of the nucleotide that carries genetic information. The bases found in DNA are adenine, cytosine, guanine, and thymine. And in a DNA, a segment of DNA that carries the genetic information is called a gene. So the genetic information is nothing but which is carried from a parent to offspring. And that particular segment of DNA which carries the genetic information is called a gene. And it is normally a stretch of DNA that codes for a type of protein or for an RNA chain that has a function in an organism. Genes hold the information to build and maintain an organism's cells and pass genetic traits to offsprings. So this forms the basis of central dogma, where from a DNA, a RNA has been produced by a process called as transcription, and from RNA, proteins are synthesized by a process called as translation. So from this, we understood the final product of a gene or a DNA is a product that is a protein. That protein may be an enzyme or it's a protein itself. But the DNA or a gene expresses to a product called as a protein. So the cycle of this process from DNA to RNA and from RNA to protein is called as a central dogma of all living things. And now, what's a recombinant DNA technology? So we have seen what's a DNA, what's a gene. And while going to recombinant DNA technology, it's nothing but it is a combination of two different DNAs or two different genes from different sources. So this combination of different DNAs can be done by a process of cutting and pasting the DNA. So in simply we can say it as cutting and pasting of DNA. The process of taking a gene from one organism and inserting that particular gene to another organism is called as recombinant DNA technology. So for this recombinant DNA technology, for doing this recombinant DNA, we need enzymes and some other accessories for this. So particularly enzymes called restriction enzyme and DNA ligase enzyme form the basis for this recombinant DNA process. And coming to history of recombinant DNA technology, it has been started in 1970 by a scientist, Hamilton Smith, at Johns Hopkins Medical School. He first isolated the restriction enzyme. Restriction enzyme is an enzyme that cuts the DNA at specific nucleotide sequence. We know DNA is very a large material, and that DNA has to be cut into fragments to form a recombinant DNA. For cutting that DNA, we need restriction enzymes, and that enzyme was first identified by Hamilton Smith in 1970. And in 1972, Stanley Cohen and Herbert Boyer combined their efforts to create a recombinant DNA. That is, they joined two different DNA molecules. And in 1978, somatostatin, which regulates the human growth hormone, was first human protein produced by recombinant DNA technology. And the basic steps that are involved in recombinant DNA technology is first, isolating the genomic DNA. Next, fragmenting the DNA using enzymes, which we call it as molecular scissors. The enzyme that cut the DNA fragments are called as molecular scissors. And the third step is screening the fragments, finding out which part of the DNA has to be combined with another DNA. So screening the DNA fragment for that particular desired gene. Next, inserting the DNA in a vector. So vector, later in slides I'll explain. And introducing the recombinant DNA to a competent host cell where it replicates and where it produces number of copies, and the particular cell will be cultured to produce multiple copies of fragments, and then we will get the transformed desired gene. Now coming to the role of enzymes in recombinant DNA technology, 6 enzymes plays a major role in producing a recombinant DNA. The first enzyme is restriction endonuclease. The second is DNA ligase. The third is DNA polymerase. The fourth is reverse transcriptase. The fifth, polynucleotide kinase. And the sixth is alkaline phosphatase. Coming to the first enzyme, restriction endonuclease, it cleaves the DNA at specific base sequence. I've told nitrogenous bases are there in a DNA. So this enzyme, it will cut the DNA at particular specific sequences. And DNA ligase, it binds 2 DNA molecules or fragments together. And DNA polymerase, it fills single stranded gaps in duplex DNA by stepwise addition of nucleotides to 3' ends. That is, it creates a new DNA molecule. Polymerase, it is used to synthesize a DNA fragment or synthesize a new DNA molecule. And next is reverse transcriptase, which makes DNA polymer of an RNA polymer. That is, from an RNA, a DNA can be created. That is done by this reverse transcriptase enzyme. And the fifth one is polynucleotide kinase. This enzyme, it adds phosphate group to the 5' end of a polynucleotide to label it or permit ligation. For taking over to ligation, this enzyme, it adds phosphate group to the 5' end of each fragment. And alkaline phosphatase, this enzyme removes the terminal phosphates from the 5' end, the 3' end, and both 5' and 3' end. The first, restriction enzyme. A restriction enzyme, it is commonly called as restriction endonuclease, is an enzyme that cuts either double-stranded DNA or single-stranded DNA at specific recognition nucleotide sequence called as restriction site. The specific sequence where an enzyme cuts the DNA is called as restriction sites. And this restriction enzymes, it cuts either the double-stranded or single-stranded DNA. We all know DNA is a double helical model as described by Watson and Crick. DNA is double helical in structure. It has two strands in helical form. So this enzyme, it cuts either both the strands or single strand at particular specific sequence called as restriction sites. And this enzymes, how it breaks the DNA molecule is, it breaks the phosphodiester bond that link adjacent nucleotides in the DNA molecule. The DNA has nucleotide units which is connected by phosphodiester bond. DNA is made up of number of nucleotides and each nucleotide is connected with one another by phosphodiester bond, and this phosphodiester bond can be broken by this restriction enzyme, so the DNA can be fragmented. The restriction enzymes recognize a specific sequence of nucleotides and produce a double-stranded cut in the end. And this cut are of two types. One is blunt-ended, the another one is sticky-ended. So while the restriction enzyme cuts the DNA, the fragment may be resulted in two types. One is it may be blunt-ended. The other will be a sticky end which can be joined easily with another fragment. The blunt end is not so easy to join with another fragment, but sticky ends are easy to join with other fragments. And going to DNA ligase enzyme, it is the most important enzyme which is used to join the DNA molecules. DNA ligase is an important cellular enzyme as its function is to repair broken phosphodiester bond that may occur at random or as consequence of DNA replication or recombination. So we know DNA multiplies. That process is called as replication. So one DNA is copied to another molecule. That is copying. The same DNA will be reproduced is called as replication. And here we are going with recombinant DNA. So during recombinant DNA also, the DNA fragments are cut and joined together. For joining, this DNA ligase is required to join two fragments of DNA. In genetic engineering, it is used to seal discontinuities in the sugar phosphate chain that arise when recombinant DNA is made by joining DNA molecules from different sources. It can therefore be thought of as molecular glue, which is used to stick pieces of DNA together. So this is also called as a molecular glue. Since it joins two fragments of DNA, it is called as molecular glue. The enzyme used most often in experiments is T4 DNA ligase, which is purified from E. coli cells infected with T4 bacteriophage. The enzyme works best at 37 degrees Celsius, but is often used at much lower temperatures to prevent thermal denaturation of the short base pad regions that hold the cohesive ends of DNA molecules together. DNA ligase is essentially molecular glue with restriction enzymes. It provides the tools for cutting and joining DNA molecules. Most commonly used ligase enzyme for any experiment is T4 DNA ligase, and for RNA, it is T4 RNA ligase. The next enzyme is alkaline phosphatase. Alkaline phosphatase is an enzyme that is involved in removal of phosphate groups. This enzyme is useful to prevent unwanted ligation of DNA molecules, which is a frequent problem encountered by cloning experiments. So applications of this alkaline phosphatase: prevention of self-ligation of vector, removal of 5' phosphate group before end labeling. So in this figure, the phosphate group has been removed with hydroxyl group. And the next enzyme is polynucleotide kinase. This enzyme is used to transfer the phosphate from ATP to the 5' end of DNA or RNA. It is also has 3' phosphatase and 2,3 cyclic phosphodiesterase activities, although of little significance. And application of this enzyme polynucleotide kinase is phosphorylation of polynucleotide. That is, it adds phosphate group to the DNA fragments. It is used in radio labeling of 5' termini of DNAs. So it has been clearly mentioned in the figure. This enzyme has added phosphate group to the 5' end of each fragment of DNA. And the next enzyme is terminal deoxynucleotide transferase. It is a template independent DNA polymerase enzyme. It catalyzes the repetitive addition of deoxyribonucleotides to the 3' hydroxyl group of oligodeoxyribonucleotides and single stranded and double stranded DNA. But this enzyme requires at least 3 nucleotides, which is used as a primer, which is used as an initiative for synthesizing. And next enzyme is reverse transcriptase enzyme. It is an RNA-dependent DNA polymerase enzyme, and therefore it produces a DNA from an RNA template. It has no associated exonuclease activity. That is, it does not have any exonuclease activity. It does not cut any DNA fragments. Its work, it develops a DNA from an RNA template. And this enzyme is mainly used for copying mRNA molecule in the preparation of complementary DNA for that mRNA. And it is a key enzyme in the generation of cDNA, that is complementary DNA. The enzyme is an RNA-dependent DNA polymerase which produces a DNA copy of an mRNA molecule. And the main enzyme is polymerase. The group of enzyme that catalyze the synthesis of nucleic acid molecule are collectively called as polymerase. And there are 4 types of polymerase enzyme: DNA polymerase 1, DNA polymerase 2, DNA polymerase 3, and DNA polymerase 4. And these enzymes are grouped into 3 groups. One is DNA-dependent DNA polymerase, that is, it produces RNA from a DNA. RNA-dependent DNA polymerase, that is, it produces a DNA from RNA. And DNA-dependent RNA polymerase that produces RNA from a DNA. And next, moving on to cloning. The term clone means producing exact copy of a parent or anything. A duplicate or a look-alike carrying the same genetic signature or genetic map. Cloning is the best application of recombinant DNA technology and could be applied to something as simple as DNA fragment or a larger sophisticated mammalian species such as humans. Molecular cloning is carried out in vitro condition where a specific fragment of DNA is isolated from an organism donor and introduced into a plasmid that replicates in a host cell, making multiple copies of DNA fragment. Cloning is nothing but taking a DNA fragment, isolating from an organism, and inserting into a plasmid, and that plasmid is transferred to your host cell where it produces multiple copies. And for recombinant DNA technology, apart from enzymes, we need some other molecules for carrying out this recombinant DNA or for producing this recombinant DNA. One such thing is a plasmid. They are nothing but they are double-stranded DNA that are usually circular and mostly found inside certain bacterial species like E. coli. And plasmids are called as vectors because it is used as a vehicle to carry our own gene of interest to combine with some other gene or DNA fragment. So since it is acts as a vehicle, it is called as a vector. Not only the plasmid act as a vector, some viral vectors are there, like cauliflower mosaic virus are used as a vector, and TA plasmid vectors are used as a vector, and so many vectors are there to carry a DNA molecule to a host cell. And plasmids, they have their unique features. They have restriction sites where enzymes can remove unwanted fragments from a plasmid, and our own gene of interest can be inserted in that, and that plasmid will be transferred to a host organism like a bacteria or a yeast. From in that organism, the inserted plasmid will produce more copies, will be transcribed and produce more copies, which can be used for recombination process. And moving on to applications. So recombinant DNA technology has applications in various fields like healthcare, agriculture, environment, and industry. Applications of recombinant DNA in healthcare. RDNA technology have a wide spectrum in improving health. It is used to treat defective gene or introduce new gene, which we call it as gene therapy. And advanced kits are available or prepared by using this RDNA technology for prenatal diagnosis of genetic disease. This we can diagnose genetic diseases in fetus itself, and a large number of mammalian proteins are being manufactured by pharmaceutical companies for use in treatment of human diseases. And it is used, this recombinant DNA technology is used in production of vaccines. Recombinant vaccines are being extensively explored, especially to eradicate infectious disease, allergies, and cancers. Example of some vaccines produced by recombinant DNA are hepatitis B vaccine, influenza and mouth vaccine, and foot and mouth disease vaccine. So one example of production of recombinant hepatitis B vaccine. So we know vaccine, it is an attenuated preparation of particular virus or bacteria itself. The organism which causes a disease, that or in that organism will be attenuated or heat killed, and that will be used as an, after processing, will be used as a vaccine. So here in hepatitis B vaccine, The DNA by recombinant vaccine, the DNA from this hepatitis B virus has been isolated, and the DNA which is responsible for producing antigenic effect has been isolated, and that DNA has been joined with a plasmid that has been taken from a bacteria. That DNA is now called as a recombinant DNA. This recombinant DNA is inserted to a recombinant yeast cell where large amount of that particular viral DNA will be expressed, and that vaccine will be purified, extracted and purified and used for curing the disease. So this is the way how an recombinant vaccine will be produced. So from an target organism, a DNA or RNA which is specific for producing that particular disease, that is, which is antigenic representative of a disease, is isolated. The gene for that antigen is isolated. The gene is cloned in a plasmid DNA. That plasmid may be taken from a bacteria or some other sources. And that recombinant DNA is now inserted to an yeast cell where it can produce multiple copies. After it produces the multiple copies, it expresses into a protein, and that protein or that enzyme or that product is used as a vaccine. And that product is extracted and purified, and it is used as a vaccine. And insulin, we know insulin, it controls the glucose level in humans. By recombinant DNA or by cloning, human insulin has been produced. That is by cloning human insulin gene in bacteria E. coli. So human insulin producing gene is isolated from the pancreatic cells, and that gene has been combined with the plasmid DNA taken from a bacterium, and this recombinant DNA is now inserted to a bacterial cell itself where it produces multiple copies of that recombinant DNA, and after some process of fermentation, this DNA will produce a product, that is human insulin, which is extracted and purified and marketed. And this recombinant DNA technology is also used for producing human growth hormone, which is of 121 amino acid to 115 Dalton molecular weight. This human growth hormone has important role in growth and regeneration or differentiation of cells and tissues. And this recombinant human growth hormone is used to treat dwarfism in children. And interferons are also produced by recombinant DNA technology. Interferons are a group of proteins that interfere with viral multiplication or replication. And monoclonal antibodies. So monoclonal antibodies are nothing but it's an antibody that are specific proteins produced by immune system in response to a specific antigen. A single clones of antibodies which is produced against a specific antigen is called as monoclonal antibodies. This monoclonal antibodies are also produced by using this recombinant DNA technology, and which is used to treat various diseases and cancer. And RDNA technology is also used to diagnose diseases Like infectious diseases are diagnosed mainly depend upon isolation and identification of pathogens, which may take several days. Developing diagnostic kits to identify pathogenic organism by knowing the organism's specific DNA sequence have provided rapid, specific, and correct diagnosis. Various diagnosis kits has been developed for many diseases like AIDS, cancer, foot and mouth disease, tuberculosis, etc. And moving on to applications of recombinant DNA technology in environment. So by rDNA technology, environmental pollutants can be eradicated. Some harmful chemicals that are not digested and degraded can be partially degradable by using this rDNA technology. One example is knockout of monodehydroascorbate reductase gene that will create resistance against trinitrotoluene. Arsenic particles are toxic and they are present in soil that is harmful for plants. The gene PVACR3 is inserted in Arabidopsis, and once the gene is expressing, it gives tolerance for plants to tolerate against arsenic and grow. So this recombinant DNA technology to produce this tolerant plant helps in producing or protecting our environment. By recombinant DNA technology, plastic degradation can be enhanced by genetically modified organisms like degrading oil spills or organic waste. One example, genetically modified strain of Pseudomonas butida is able to degrade chemicals in oil spills, and bioindicators has been developed. Some bacteria has been genetically modified and used as bioluminescence that gives off light in response to even small chemical pollutant if it is present. Applications of recombinant DNA technology in agriculture. rDNA technology can be used for insertion of genes in plants, not only from related plant species but also from unrelated species like microorganisms also. And this rDNA technology is used to produce transgenic plants, used to develop root nodules in cereal crops, and develop C4 plants. An importance of rDNA technology in plants is to produce transgenic plants with higher yield, higher nutritional values, increased stress, resistant to stress and pest, and it is also used as a source of biopharmaceuticals. So for production of high yields in plants, we can go with recombinant DNA technology. For higher nutrient values, we go for recombinant DNA technology. And for increased resistance to stress and pests, we go with recombinant DNA technology. Some examples. Researchers at Japan's National Institute of Agrobiological Resources added maize photosynthesis gene to rice, and they got the result increased yield by 30%. And Mexican scientists have genetically modified plants to secrete citric acid from their roots, In response to slight increase in acidity, minerals bound to soil particles and are released and made available to the plants. Stress tolerant plants, plant resistant to environmental stress. DNA technology is used to develop crops that can tolerate abiotic stress. Genetically modified tomato and canola plants can tolerate salt levels up to 300% greater than normal. Insect resistant plants, the crygen, which is popularly known as Bt genes from the bacterium Bacillus thuringiensis, are isolated, and it has been modified. And that modified gene has been used in recombinant cotton, rice, maize, potato, brinjal, cauliflower, etc. This recombinant Bt genes has been used to produce recombinant plants like cotton, rice, maize, potato, brinjal, cauliflower, etc. The next is drug resistant plants. Plants are modified to produce resistance against diseases. One example is tobacco plant was first modified to produce resistance against tobacco mosaic virus. Application of recombinant DNA technology in industries. Traditional industrial microbiologies merged with molecular biology to yield improved recombinant process for the industrial production. Primary and secondary metabolites, protein biopharmaceuticals, and industrial enzymes are produced by using this recombinant DNA technology. Important primary metabolites like amino acids, nucleotides, vitamins, solvents, and organic acids are produced by RDNA technology in industries. And these primary metabolites are used for growth of cells and organisms. And its vitamins. Vitamin B12 is produced commercially by direct fermentation utilizing the fungus Aspia gospi. Vitamin B12 is produced by direct fermentation using Streptomyces species also. And vitamin C is produced by using Glucanobacter oxidans. And beta carotene is produced by Phycomycetes family. And amino acids like glutamic acid, lysine, phenylalanine, valine are also produced by cloning gene using the vector PBR322. And amino acids like L-glutamate and L-lysine is mostly used as feed and food additives. And moving on to transgenic plants. The genetically modified tomato called as flavor saver tomato, which has long shelf life, is produced by this recombinant DNA technology. And this tomato was the first genetically modified tomato and commercially available. It was produced by the Californian company Calgene and submitted to the US Food and Drug Administration in 1992. And after this, 1995, BT potato was approved safe by the Environmental Protection Agency. So how this flavor saver tomato has been produced? So normal tomato, it will rotten easily and quickly, whereas this, it will take about more than 3 weeks to rotten. And BT cotton, which is genetically modified cotton, which is resistant to pests, and golden rice, genetically modified to contain large amount of vitamin A, that is beta carotene. And varieties of golden rice has been produced by this recombinant DNA technology. Next, moving on to transgenic animals. It's a miracle in genetic engineering. You can see this a frog through the skin, how organs grow, how cancer cells starts and develop without disrupting the frog. And this is a genetically modified frog. And the glowfish, which was first genetically modified animal to become available as a pet, is a natural zebrafish, which has genetic information from bioluminescent jellyfish added to its DNA. A cross fertilization between a lion and dog, Dolium. The dolly, the sheep, it is the first recombinant DNA animal produced. From one sheep, a nucleus or DNA is taken. From the another sheep, egg is taken, and these two were given to an surrogate mother or foster mother, where it produces the cloned lamb, which is named as Dolly. Dolly is the sheep, is the world's most famous clone. It was born on 5th July of 1996 to three mothers, that is, one provided the egg, the another DNA was taken from the another, and the third, it's carried the cloned embryo and given birth to Dolly. And next is Zorse. It's a cross between a zebra and a domestic horse. The crosses were originally done in England, Africa, to try to produce a domestic horse-like animal that was resistant to diseases spread by the fly in Africa. Even though this recombinant DNA plays major role in all the fields like agriculture, industries, medicine, environment, but there are some ethical crises by this recombinant DNAs. We have ethical crises. So not all the recombinant products are available. Once the ethical clearance has been done, the product will be available in the market. The basic of this recombinant DNA is combining a gene of interest that may be taken from a source of plant or animal or a bacteria or any other organism, combining it with another organism's DNA and to produce a new combinant of DNA which will express to a protein. So here This central dogma plays an important role where a gene is transcribed to RNA and it is translated to a protein, which is a product. For example, for producing a transgenic plant, a gene from, for example, a gene from a stress-tolerant plant will be taken. It will be inserted to a normal plant through a vector called as plasmid or any other vector, and in that plant it will express that particular product, that is particular gene, where it will become tolerant to that particular stress or anything else. So this is the way how a recombinant product or recombinant DNA is produced. So thank you for giving me an opportunity, sir.
Hello. Good evening, Bedkar sir. Good evening, madam. Wonderful presentation what you have done today. Really appreciated because something we are going in a very nuclear, very central this place. Nowadays, the technology is advancing. We are talking about the robotic technologies and robotic medicine and nuclear medicine and so many things in our field. And I think that is a part what you are talking today. And that is not applicable only for the human being, but that is also applicable for the plants and various kinds of bacteria. So we can utilize it at various ways and various places. That is wonderful. But what are the disadvantages of this kind of, when we are doing this kind of, we are using this kind of technologies? Can you identify any kind of disadvantages? Because we are saying cloning is also one part of a human being, depends upon this only. So what are the disadvantages? Thank you, madam.
Yes, sir, we have disadvantages of. Sir, can I answer? Sir, this recombinant DNA technology, it goes against the law of nature. We have some religious beliefs, we have some laws of nature, and this goes against the law of nature. This forms one crisis, ethical crisis. And one more thing is, sometimes even if a vaccine, prepare vaccine by attenuating or heat killing or some other only. So in some cases, very rare cases, it may revert to its original thing. So, but in the recombinant, that can't be reverted. It may give some adverse effects. Even if a plant is tolerant to a stress, that plant, recombinant plant, may give some other effect, like it may, if we are producing a stress tolerant plant or if we are producing a high yield plant, that may give some other effect in producing some other metabolites in that particular plant. It will reduce or it may increase the product rate and so on.
Yes, madam. Thank you. What has happened, when we are cultivating the hybrid vegetables and other things, that kind of nature where they have lost their what we can say natural essence, and that is how nowadays what we get of vegetables and fruits, they sometimes they don't have taste, but their appearance and their sizes are big, and only the the natural, the flavor and that essence has lost. So by doing using such kind of technologies, one has to be very clear, and our conscious should be there, when we are doing such a kind of activities. And biggest question is of cloning, because if suppose we can clone people, human beings, and that will create a big chaos and chaos in the world. Thank you very much, madam, for your wonderful presentation.
Yes, sir. Thank you. Thank you, sir. Thank you, sir. Thank you for giving me this opportunity. Thank you, sir.